Okay, so it's wonderful to be here today. This is our first Cannabis Grand Rounds of 2022, and we're very honored to have Dr. Tony George with us. Before we start with the introduction, I just want to give a couple of brief announcements. The first is that we will be skipping next week's faculty meeting. It was scheduled for March 24th, but because we have Dr. George here with us today, and then we have a seminar on March 31st, which is Thursday, we're going to skip our faculty meeting next week. So just remember to save the date. You all received an announcement for our March 31st seminar with Dr. Elisa Pabon, who's a postdoctoral fellow at the University of Chicago. We're so excited to have her join us for a seminar over Zoom on the 31st. And then also keep in mind that our next faculty meeting is April 21st, and we expect to have some short student presentations during that faculty meeting. Also, the CMCR San Diego will be having their annual event on April 21st and 22nd, Thursday and Friday. So we're hoping that you'll attend that event, but also maybe get away for 1 hour so you can join our faculty meeting on April 21st. So those are some general announcements. So for today, if you have questions, I'd like to direct you to putting your questions in the Q&A box. And also, if you would like to see closed captioning, you can click on the live transcript on the bottom bar all the way to the right, and you should be able to see a live transcript. So today, I'm so happy to introduce Dr. Tony George. He's the professor of psychiatry at University of Toronto and at the Center for Addiction and Mental Health called CAMH. He is the senior clinician scientist of the Addictions Division and the scientific lead for addictions research. Dr. George's main research and clinical focus lies in the program he directs, which concentrates on understanding the biological basis of addiction, comorbidity, and serious mental illness with a focus on schizophrenia. And you may have read about his most recent publication that he'll be talking about today, which investigated the effects of Rtms, repetitive transcranial magnetic stimulation, in patients with schizophrenia and cannabis use disorder. It was really fun to see this paper highlighted in many media sources, and I'm sure that you also saw some of this attention as well. Dr. George has held many prestigious leadership positions at University of Toronto. He was the inaugural holder of the chair in addiction psychiatry. And at CAMH, the Center for Addiction and Mental Health, he served as chief of schizophrenia division for nearly a decade. And during this time, he also served as the medical director of the complex mental illness program. And most recently, he served as the chief of addictions at CAMH. Dr. George's research is supported by the Canadian Institutes of Health Research, CIHR. Some of you may have reviewed for CIHR in the past. His research is also supported by the CAMH Foundation and NIDA, National Institutes of Drug Abuse. Dr. George has close to 300 peer-reviewed publications, many of which really concentrate on his primary research interests, the biological basis of addiction, comorbidity, and serious mental illness. Dr. George is a fellow of the American College of Neuropsychopharmacology, ACNP, and he has served as deputy editor of ACNP's journal, Neuropsychopharmacology, for nearly a decade. And it's very exciting to announce that he will be the co-editor-in-chief of NPP starting in 2023. So that will be great. Congratulations, Dr. George. And just on a personal note, I've talked a lot about Dr. George's leadership positions and research, but I want to say that many of you know him as an incredible mentor and colleague. I first met Dr. George at my first NIH study section, which was very intimidating situation for me, first time here in the study section. And Dr. George really took me under his wing. And I was going through an interesting career time at that point where I was trying to figure out where my next step would be. And Dr. George started the conversation with me, and he was always willing to have a phone call, have a Zoom, talk to me about different opportunities and where to get where I wanted to be. And I know personally that he's also very committed to mentoring graduate students and postdoctoral fellows. He has introduced his students to me, and we have developed mentoring relationships in that way. So, for me, this is a real personal pleasure to have Dr. George here to speak, not only because of his excellent research, but also because of the outstanding mentorship that he has given to me, as well as other students and junior faculty. So on that note, Dr. George, I'd like to invite you to talk about the mechanism of cannabis effects in people with mental illness, translation to treatment. Thank you.
Well, thank you, Ziva. Thank you so much for inviting me. It's been, let me see if I can just share my screen here. I've rehearsed this a few times, so hopefully I'll get it right this time. There we go. But Ziva, you've been, someone near and dear to me, and we've started to collaborate now. And I realize I have so many friends at UCLA. I was telling Ziva that, there's like 60 some people. I only have 65 friends, so they're obviously mostly at UCLA now. So, but I'm going to talk to you about something that's near and dear to my heart, which is cannabis in people with mental illness. And so let me just fly right ahead here. Just a few disclosures here. Just start out with, this is something from our West Coast. This is from British Columbia. This is Gabor Mate, and some of you might have heard of this book that he wrote, mostly about heroin addicts in Vancouver. It's called In the Realm of Hungry Ghosts. But if you read it, you realize, when you're talking about people with mental illness, you use drugs, they become, truly addicted, enslaved, and they become like the hungry ghosts, like the heroin addicts that he described, with compulsive, impulsive behaviors that get them into serious trouble, and often it's their demise is related to their drug use. Well, that certainly happens with people with psychosis who use cannabis, and mood disorders even as well. And I just showed this slide, the causes of disability in the Western world, the US and Canada, because number one and number two are mental illness and addictions and alcohol and drug use disorders. So if you add them up, they're 40% of the disability in Western society is attributable to mental health plus addictions. And that's probably an underestimate. And some of you know this data came from President Bush's New Freedom Commission on Mental Health almost 20 years ago. Of course, one of the auspices of that perhaps was to defund mental health and addictions, but as we know, that didn't happen, thank heaven. So, but moving forward, I like to go right back to the beginning, and I told Ziva that I'm a Michigander, and unfortunately, I'm so narcissistic, I'm showing a picture of me when I was 18 months old, but it also tells you why I do what I do. This is me on the grounds of the Ypsilanti State Hospital in Ann Arbor, or Ypsilanti, Michigan, holding a pipe. My dad was a psychiatrist. He was a psychiatry fellow there, and he had a warped sense of humor like me. And so he had me, extolling the benefits of nicotine and tobacco. And ironically, that was my first area of research. Fast forward to 2006, this was the team that I had at Yale at the Connecticut Mental Health Center. We had the auspicious name of the Program for Research in Smokers with Mental Illness, or PRISM. Every time one of our research subjects came to the front door of the Connecticut Mental Health Center, they'd say, "I'm here for the PRISM program." And or anytime a forensic patient showed up and said, "I'm here for the prison, I'm here from prison," they used to send them to my program. And of course, we'd have to redirect them to the other side of the center, to our forensic colleagues. But I'm proud to say that the back row there were all incredible women scientists who work with me. Two of them are psychiatrists, two of them are academic psychologists, and two of them are still at Yale doing incredible work. And the three guys in the front, including Mark Potenza, we turned out to do nothing at all. So complete waste of time. So, but anyway, that was my start, and it sort of just outlines kind of what I tried to do with tobacco and mental illness that sort of set the stage for the cannabis mental illness work. So this are two of my people, then Christy Sacco, who is my neuropsychologist, and still on the Yale faculty, and Angelo Termine, who was my lab manager. And they had this, really interesting result that was published in the Archives of General Psychiatry, now JAMA Psychiatry. And what it showed here is that looking at cognitive enhancement with this frontal lobe related task that we've been studying for the last 25 years called visuospatial working memory, or the DOT test, which comes from Richard Keefe's work at Duke. And what we were able to show is that in schizophrenia, they had this incredible enhancement of spatial working memory function. But compared to controls, it was just the opposite effect. In fact, our non-psychiatric control smokers, when they smoked cigarettes, they actually impaired their spatial working memory. And then we used the only available nicotinic antagonist at the time, mecamylamine, which is pretty non-specific, and it completely blocked the enhancement in people with schizophrenia, and it probably did nothing in the controls, or maybe even enhanced it. So there was these diagnosis-specific differences in the effects of smoking and nicotine. So by blocking those nicotinic receptors, we took out the operative ingredient that was stimulation of nicotine's receptor, the nicotinic acetylcholine receptor. So this study was, kind of got me on my way. It was my first night at Grant, already having, and I wasn't going to get into this, but these were three fantastic investigators. Sherry McKee, of course, many people know, tenured professor at Yale from London, Ontario originally, did some great work on smoking topography and showed diagnosis-specific differences. Andrea Weinberger, who was a postdoc of mine, now a professor at Yeshiva in New York City, looked at rewarding effects. And then Margot Fonder, who was an undergraduate student with me at Yale, now a dermatologist at Brown in Providence, Rhode Island, looked at cue reactivity. And again, nice evidence of these diagnosis-specific differences in smoking effects in schizophrenia versus controls that you could bring out using an antagonist to sort of to accentuate those differences. And we're not really sure why there are those differences. This was a model we put together at the time, kind of looking at, biology meets, genetics and environmental factors. We really focused on prefrontal mechanisms because of the spatial working memory findings. We tried to relate this to, the higher rates of smoking in people with schizophrenia, their more likelihood of not quitting smoking, and their frequent relapse. And so this was a nice model that Vicki Wing, a postdoc at the time, put together. We've actually adapted this in a chapter in the new upcoming U.S. Surgeon General's report on tobacco and mental illness. But then, I'll just, as a caveat, this is George Box's quote: "All models are wrong, but some are useful." And so the proof was in the pudding. And what we did at the time was, this is Sarah Dolan, who was a graduate student with me at Yale, now a professor and vice chair of research at Baylor in psychology, and Taryn Moss, who was my first graduate student in Toronto, is now a practicing clinical psychologist in Toronto. And what they showed is that in people with schizophrenia who tried to quit, those who had preservation of prefrontal executive function and better working memory, better frontal executive functioning on the trail making test, part B, and the digit span backwards, all classic tests of frontal executive function, those people actually quit smoking compared to ones that had deficits. So it was sort of an early start for me in understanding, how the prefrontal cortex and cognitive executive function were important to address, particularly in a disorder like schizophrenia, where there's well-known deficits in prefrontal function. So now to the present day. So this is actually a picture of a sign on a telephone pole at Queen and Ossington in Toronto, where CAMH is, and it's advertising 2-hour shroom and weed delivery, and the service is known as the Loose Moose Delivery. Of course, they restrict it to age 19. Of course, weed is legal in Canada like it is in California. Ironically, there are 24 cannabis shops within a stone's throw of the front entrance of CAMH. Completely coincidence, I'm told. And so, you know, these are our strengths and failures in drug policy, I think. We're trying to do some psychedelic studies in the new center I work in. It's taking almost a year to get secure, you know, the FDA and Health Canada approved supply. And I keep saying to our director, you could get it in 2 hours, but apparently our IRB would not accept that. So, but anyway, that's the, and you know, of course, one important point is I don't know how much penetrance legal cannabis has had in California, but there are statistics now in Canada that legal cannabis only makes up 46% of the market. So unfortunately, the illegal industry, as you see here, is still chugging along strong. So this is my pandemic productivity I'm going to show you next, and sort of emphasize what we tried to do in sort of framing what we do. So one of the questions we wanted to take another look at, just because we had so much time because of the pandemic, like Ziva was telling us, you know, starting up clinical research, you know, in the post-pandemic, if we can call it that era, still has its heartaches. And so one of my students, who is going to figure prominently in this presentation, Miriam Soaku, when she was a first-year graduate student, all of 18 months ago, when she started out, I asked her to take a look at this question: what are the behavioral effects of cannabis in otherwise healthy people? And so she did this systematic review, very simple. How many studies looked at positive effects or negative effects of cannabis, as may be the case, and how many of them did not show anything? And anything where we show 80% or more of positive studies or negative studies, if you will, we classified as the highest rating, a 5, and if it was less than 20%, it was a 1. So you can see the classification schema down below, which, you know, is there for replication. And it turned out, at least in this look at the literature, that the only ones that met that 80% threshold were psychosis, surprise, surprise, cannabis and psychosis, and psychosocial functioning. 7 out of 8 studies on psychosocial functioning. You know, motivation fell a little bit below that. IQ kind of mixed. You know, lots of fair play about declining intelligence in cannabis using kids. But it was just a reminder to me that we need to keep taking a look at these things, because there's a lot of hype about cannabis and what it does, and its negative and maybe even positive effects. And so, you know, it's all about keeping us honest. And so Miriam, with this undergraduate student, Rachel Beater, put this very nice paper together in Frontiers in Psychiatry that was then, you know, cited in Psychology Today, which was really cool. This is something that many of you know. This is looking at the average age of cannabis use onset in kids and adolescents. And at the time we did this report, this was the report on cannabis in youth for the Canadian Centre for Substance Use and Addiction. I've been affiliated with them for the last 15 years. They're Canada's clearinghouse for drug and alcohol use information. They do knowledge translation, capacity building. And we put together this report on this topic. And at the time, the average age for first use of cannabis was 15 in Canada. It's now 13.3. So even since we legalized in 2018 and our legal age is 19, that age of first use keeps getting lower and lower. And that's relevant actually, because the onset of different psychiatric disorders falls right in the middle of that. So you look at some of the big ticket ones like schizophrenia and serious mood disorders, they typically happen not too long after that, particularly schizophrenia. So this is important, understanding these interrelationships between cannabis and mental illness. Another look at this is Grace Volkoznik, who's a senior undergraduate student at McGill University in Montreal. And 2 summers ago, I asked her to look at another question. So I asked her, what are the effects of cannabis specifically on psychosis, hypomania, and depression. And she pulled out 47 studies. And it turned out that almost all the ones in psychosis were negative. That is to say, there were very deleterious effects of cannabis. In fact, there were 6 experimental studies where they administered THC or cannabis under laboratory settings, including some Cyril D'Souza studies from Yale. And in almost all cases, psychosis was the result of cannabis administration in healthy people. But only 4 studies in people with bipolar disorder or evidence of hypomania or mania, and a few more studies in depression, but much more mixed picture. There were some experimental studies that showed the positive effects or the deleterious effects of cannabis in people with depression, but not so much the case or a mixed picture with cohort cross-sectional studies. So this was Grace's first publication, and so very proud of the work that she did 2 summers ago. And then finally, this is Carolina Kozak's work, and I think you know the person on the bottom there. She was a wonderful collaborator on this study. Thank you, Ziva, especially for suggesting your journal, American Journal of Drug and Alcohol Abuse. Turned out to be a great place to publish it. What Carolina and Ziva and our team showed was that knowing that there's a sex difference, a 2-to-1 male to female ratio in the general population, might be even lower than that, but at least from the data we found, it was 2-to-1. If you looked at different psychiatric disorders, they were sort of all over the place. The strongest male-female difference was in schizophrenia and unipolar depression. So 2.2 to 3 or 2.6. But then other disorders, personality disorders, bipolar disorder, opioid use disorder were much less, closer to 1. So there were some methodological challenges in doing a systematic review and meta-analysis like this, but very interesting to look at how mental illness can be an important factor in modulating gender differences. So let me move now to some of the experimental work that we've done with cannabis and mental illness over the past couple of years. So this is a general study design for our approach methodologically. And we have a smoking room at my lab at CAMH where we can actually do smoking, tobacco, and cannabis smoking studies. The province has actually given us carte blanche to do almost any kind of challenge studies in those rooms. We've mostly done cannabis and tobacco to date. But when we're talking about people with mental illness, of course, from a research ethics point of view, doing challenges is going to be a little trickier. So I grew up in an environment when I was a junior faculty member at Yale where John Crystal and his group were doing ketamine challenges pretty repeatedly. I know UCLA has a history of doing this, but we decided to take an opposite approach. We decided to take away cannabis from people who were physiologically dependent on it and get them off it long enough so that we could study its effects post-withdrawal on clinical symptoms and cognitive function. And so what we were able to do is study these people, enroll them, characterize them well, test them at baseline before they went into abstinence, extended abstinence for, in this case, 28 days. And you may ask, how do we get them abstinent for 28 days? Well, we use the most powerful behavioral technique known to not, have to use a drug and do this in a purely behavioral sense. Of course, we paid them. So the Jerry Maguire, show me the money, or the Donald Trump money, money, money approach. And so what we did is we said, if we could prove biochemically with a drug screen, urine drug screen, that you're abstinent, we will give you $300. But if you're not meeting our criteria for biochemically verified abstinence, you won't get anything, except we pay them for every visit, obviously, right? And then actually after we gave them the reinforcer, we followed up with them 1 month later to see if they relapsed. So, and actually I showed this slide last when I was at the AAA conference in in San Diego back in 2017. And one of the comments on the evaluation was, great talks, like Dr. George's talk, except when he reminded me that Mr. Trump was our president. So anyway. So let me show you some of the results that we found. So this is my former grad student, Aaliyah Lukács, proudly showing us her master's degree that she got for doing this study. And what Aaliyah did was she asked the question, what would 28 days of abstinence from cannabis do to the symptoms of people who have major depression and cannabis use disorder? So using this paradigm, she was able to get about, I think out of the 14 people we studied, she was able to get 10 or 11 of them to quit cannabis, biochemically verified. And we ended up counting 11 of them because there were some discrepancies in the baseline diagnosis. We had to exclude some people. So she had 11 people going through this with major depression. And what she found was a 50% reduction in depression symptom severity on the Hamilton Depression Rating Scale 17 item, the standard in the field, or one of the standards in the field. So that was quite profound. Obviously, cannabis was not an antidepressant. You take it away, their depression gets worse, okay? We looked at this a little bit further. This is Sakina Rizvi, who is a collaborator of ours. She's on Mariam's graduate committee, program advisory committee. She's an expert in anhedonia and collaborates with Diego Pizzagalli at McLean, looking at aspects of anhedonia, the inability to feel pleasure or amotivation. She uses much more sophisticated measures than we do. This is a self-report known as the SHAPS, which is a standardized measure of anhedonia severity. And to our delight, these patients who had fairly significant anhedonia on the SHAPS had an 80% improvement in their anhedonia with quitting cannabis, with biochemically verified cannabis abstinence. So most recently, and this paper is under revision now at the American Journal of Addictions. Larissa's, the AAAP, where Larissa Mooney is president, this is their journal. And what we're able to show is that working memory task that I told you about, spatial working memory task, has a significant improvement with cannabis abstinence, huge effect sizes. And this is Rachel Rabin, my former PhD student, and her twin sister, Jenny Rabin, who's a neuropsychologist, who was at Sunnybrook Hospital in Toronto. As I told you, Rachel is at McGill, and they brought their tremendous expertise to this study. So very proud of Miriam and the Rabin sisters for pulling this paper off. Let me just tell you, this is Miriam again. I told you she'd be featured a lot in this presentation. And so Miriam's PhD thesis is to do a controlled study of this process and look at sex and gender differences in a much larger sample. So what Miriam is currently doing, she's just enrolling the first subjects in the study. She's got about 4 or 5 already. And she is randomizing them to either the contingent reinforcement active condition, where they'll be told you'll get $300 if you quit, or the treatment as usual or control condition, where people will just try to quit with behavioral counseling, just standard less than 15-minute behavioral counseling, which is what we offer as the standard of care in our clinic. And of course, that last group, the non-contingent reinforcement group, is the straw man, but it is the necessary control, the time dependent control that we need to verify these effects in a larger sample. So Miriam, hopefully in a few years, will have some very interesting findings that we'll be able to talk about. So let me move on. Oh, and let me just tell you, this is Justine Rodas, another grad student who works with me, and her co-supervisor, my colleague, Ahmed Hassan, who studies behavioral interventions in complex people with opioid and other drug disorders. And what they're looking at is a very innovative study where they're going to apply this 30-day abstinence paradigm in post-traumatic stress disorder. They call it the CAN-POST study. They're actually taking it one step beyond. So they're using the randomized design that Miriam's using, but they want to extend it out to 12 weeks instead of 4 weeks like we did. So they're actually going to pay people progressive amounts of money, $200 at 1 month, $300 at 2 months, and $400 at 3 months to see what extended abstinence from cannabis does to symptoms and cognition in PTSD. So another exciting study using this basic paradigm that we have developed. So let me move on to the schizophrenia part, and I'll try to do this in about 20 minutes. I'm going to talk to you about 2 main studies that we've done and sort of lead up to the neuromodulation stuff that we've done most recently. So this is Darby Lowe. She was actually part of the sex differences paper that Ziva was on, and she was my master's student. She'll come up later in the presentation on the neuromodulation stuff. And so Darby wrote a really nice review that's been cited now in the past 2.5 years over 110 times, according to Google Scholar. And, you know, this just makes the point that people with serious mental illness have very high rates of cannabis use and cannabis use disorder. So if rates of CUD in the general population of the US around 3%, this is Deborah Hazen's data from Columbia, it's much higher. It's probably in the range of 8 to 12%, in some samples as high as 25% in people with mental illness. And at least in schizophrenia, there's a real male to female difference, a real sex difference. You see about 2 to 3 to 1, like I told you, of people who are men having cannabis use disorder versus women. We know there's a very negative impact on the course of the disorder, more hospitalizations, poor prognosis overall, and increased treatment needs. And so, you know, schizophrenia, which still takes up 20% of beds in America and Canada, this drug dependence is a serious problem that gets them into big trouble. I just show you, you know, I'm not a geneticist, but this is someone we started to collaborate recently. This is Emma Johnson out of Washington University in St. Louis. And this is just a very nice slide showing there's a very sophisticated way that you can show or explain genetic explanations for the comorbidity of cannabis in people with schizophrenia and the different ways that you can do that. And this is a paper that she recently published in Addiction, using a very sophisticated statistical genetics approach to show the interrelationship between cannabis and schizophrenia, and also tobacco, which is very common in these patients. And I'll show you some cool interrelationships that we've uncovered in our neuromodulation study between cannabis and tobacco and schizophrenia. This is Cyril D'Souza, who a lot of people know very well in both the schizophrenia and cannabis fields. Cyril led a really nice consensus review and guidelines in the World Journal of Biological Psychiatry with a whole bunch of us. And this is just a lovely figure from that, showing that the more cannabis you use in terms of amount and potency, the more likely it is you're going to get psychosis. It's a greater risk of psychosis and schizophrenia. So that review should be out very soon in the World Journal of Biological Psychiatry. This is just a reminder to me, again, why genetics is important. And this is Avram Caspi's data that was published, 18 years ago now in Biological Psychiatry. Basically just showed that these polymorphisms in the catecholamine O-methyltransferase gene that govern dopamine metabolism in the prefrontal cortex can either make you more vulnerable to psychosis, i.e. the Val-Val genotype, where there's rapid dopamine metabolism, or the MET-MET, which is slow or low dopamine metabolism that stabilizes dopamine levels and reduces chance of cannabis-related psychosis. So this is a very complex and interesting field. So now this is Rachel Rabin, who's now an assistant professor at McGill, my first PhD graduate in Toronto, and she did a series of very programmatic studies during her PhD where she showed, fairly high rates of cannabis abstinence in the schizophrenia population compared to non-psychiatric controls. It was 42% in the patients versus 55%, actually not statistically significantly different. She showed selective improvements in verbal learning and memory, and that's the inset graph there, and improvement with the first 28 days of abstinence. And when they relapsed, spatial working memory got worse. And so that was a beautiful finding. I think it was a beautiful finding. I'm biased, but it was published in NPP. So we were very happy about that. And then interesting, no changes in psychotic symptoms, but a reduction in depression and a compensatory increase in tobacco use in people with schizophrenia, but not in controls during 28 days of cannabis abstinence. So we're very happy about that group of studies, and I think it taught us a lot about the further interrelationships between cannabis and psychosis, and including cognitive and clinical function. We do a lot of imaging at CAMH, and this is Isabelle Beaulieu and her student Claudia Paluga, who's on the Canadian national tennis team. And they're looking at this ligand for fatty acid amide hydrolase, which is, of course, as many of you know, the enzyme that breaks down anandamide, the endogenous endocannabinoid. And what they've shown, using a short-term abstinence paradigm, managed to convince a few of my colleagues about the importance of contingency management. So that's what they've been doing. And they actually show restoration of FAW binding. So it suggests that when you take away cannabis, it actually makes you more likely to have FAW, which is a good thing. So Isabel's first paper in biological psychiatry showed that with cannabis treatment, you could restore FAW levels. And so there is this very interesting study that Cyril D'Souza's done using a FAW inhibitor now as a potential pharmacotherapy for cannabis use disorder. And the first study published in Lancet Psychiatry a few years ago was positive. So Claudia and Isabel will be presenting this at the Canadian Cannabis Conference in a few weeks. But let's get to the neurostimulation stuff in the last few minutes. Ziva told you that paper of ours came out just recently. This is Caroline Voss, who started off our neuromodulation studies at CAMH. When I came to CAMH about 15 years ago, Jeff Daskalakis, who's now, as you may know, is the chair of psychiatry at UC San Diego, really got us going with this. He's been doing this for many years. He's, of course, a superstar in the field right now. And, it's been used for a lot of different disorders, of course, approved for depression. It's been used to treat auditory hallucinations and schizophrenia, in other neuropsychiatric illnesses, including Parkinson's. We use high frequency, usually greater than 10 hertz. We use 20 hertz. And I'll tell you a little bit about the paradigm that we use. But to start off, we ask the question, this is our general question, can high frequency RTMS directed to the frontal lobes target comorbid addictions in people with schizophrenia? So that's been our sort of overarching question. And of course, this is Carolina, my second PhD student after Rachel. She's now in the private sector. She led the cannabis study the RTMS study of cannabis in schizophrenia. And this was her cognitive neuroscience model that predicted that if we stimulated the prefrontal cortex bilaterally in a 1 to 2 centimeter penetration, it would ultimately target associated reward structures and lead to a reduction in substance use. Fairly simple model. I've taken a pretty simple-minded approach to this. We published a review a few years ago in the American Journal of Addictions. This is Alex Coles, who still works with our bigger group, showing that essentially if you target bilateral prefrontal cortex or even unilateral prefrontal cortex, that is the way that you can actually see positive effects of different modalities, particularly RTMS and TDCS and tobacco, alcohol, but other substances, including cannabis, really hadn't been researched. So this is the great knowledge gap that we have in the field of neuromodulation right now. very little with deep brain stimulation. I'm actually part of a group at Sunnybrook Hospital in Toronto that's using deep TMS, deep brain stimulation, deep brain stimulation to treat alcoholism by stimulating bilateral nucleus accumbens. And our first report on that should be coming out soon. So we first applied this to tobacco smoking and specifically tobacco craving. And we used a 28-day paradigm that had been shown to improve working memory on the NBAC test that Jeff and his colleagues had done in their studies. And lo and behold, when we did this, and we had a small grant from CIHR to do this, Vicki was able to show that in a paradigm where we briefly deprived smokers with schizophrenia of their cigarettes and could increase tobacco craving, that is the dotted lines that go up, applying RTMS would suppress that, active RTMS, that is. The dotted lines are actually sham RTMS. So that was interesting. We could suppress tobacco craving. But being ambitious, we got a bigger Canadian Institutes of Health CIHR grant, and we decided with Jeff and other people's advice that if we did really intensive stimulation instead of once a day, twice a day for 5 days, we could use our laboratory model we use with drugs like mecamylamine to look at this in a much more shortened paradigm. Well, we were completely wrong. And Carolina led this study as well as part of her master's thesis. We showed no effects with our 5-day stimulation paradigm. There was no effects on craving in either patients or matched healthy controls. So back to the drawing board. And so I'll skip some of the steps, but in the interest of time. But in the last 5 or 10 minutes, let me tell you about this cannabis brain stimulation study we call CAN-STIMULATE. So it's the cannabis schizophrenia treatment with intensive magnetic stimulation, or CAN-STIMULATE. So not my best acronym. PRISM was my favorite acronym. But it was supported by NIDA, and we had a wonderful collaborative interdisciplinary team at CAMH that, once again, Carolina led. And let me show you what we found. So this is the group that did this. This is the inclusion exclusion criteria. These were mostly men, ages 18 to 55 with DSM-5 cannabis use disorder and with schizophrenia, full scale IQ greater than 80. They had to have a positive urine screen for THC, no metallic implants on the exclusion criteria. This was, I think, a very methodologically rigorous study. And I'll show you some of the evidence for that. This was the paradigm, simple randomization to either active RTMS or to sham. And the refinement with this is that we acquired the sham coil that looks like the active coil. It sounds like the active coil. In fact, it buzzes like the active coil, but it doesn't deliver a magnetic field to the target area, in this case, the prefrontal cortex. So 20 hertz daily for 30 minutes, 5 days a week for 4 weeks, or sham. And one of the other methodological refinements is anyone who studies people with schizophrenia know that there's high rates of dropout in these trials. So we were worried about that. And the R21 only supported 36 to 40 subjects. Of course, COVID came right in the middle of it. So we ended up with about 20 subjects randomized. And to make sure that they stayed in the study, we paid them increasing amounts of money. It could be gift cards, it could be cash, depending on what the patient, families, and case managers agreed to. And we escalated those payments. And I'll show you the results of our retention shortly. But that was instead of reinforcing abstinence, we reinforced RTMS session attendance. This is Daniel Bloomberger, the new director of our brain stimulation center called Temerty, the Temerty Center. He took over from Jeff. He's now a full professor at U of T in psychiatry, great guy, very easy to work with. He provided all the technical support. He provided all the equipment and the trained personnel. did many of the resting motor thresholds himself, and ensured that the study was rigorously done from the neuromodulation point of view. This was just our consort diagram. I'm really into consort diagrams because that's my role as deputy editor, to champion the clinical trials and the clinical studies, the human studies. So I got to know this pretty quickly, even though I knew nothing about it. But what was, maybe a little bit disappointing is that we only screened 100 people for this study that was interrupted by COVID. We enrolled 24, 5 dropped out right off the bat for various reasons. We randomized 19 people, 10 to the sham group, 9 to the active group. We had pretty good retention. We only lost 2 people along the way. We even did a 1-month follow-up. Just a little bit about the demographics. These were people mostly with schizophrenia, generally quite young, actually. Many came from our early psychosis clinic, mostly on 2nd generation agents. IQ was actually a little higher than normal. They were, high school, post-high school educated, very stable from a clinical point of view on the positive and negative scale, symptom scale of schizophrenia, low depression scores on the schizophrenia specific scale, the Calgary depression scale. Smoking just under a gram typically of cannabis a day. We did this point of care test in addition to our GCM-SMS gold standard. They had pretty moderate levels of carboxy THC, cannabis metabolite in their urine. QDIT, which is the an analog of the alcohol use disorders identification test for cannabis, moderate level of impairment, et cetera, stable baseline, stable outpatients. So this was the first result, and I'll be done shortly, I promise, Eva. Retention was really good. We lost 2 people in the trial of 19, and they were both in the sham group. Of course, the Kaplan-Meier statistics showed no difference because the small sample, but we look at this as the glass is half full. This paradigm with reinforcing RTMS session attendance did really well. And actually in a study that we're planning, Steve Martyr is part of that. And Steve said, I don't know if Steve is there, but he was very happy about our retention numbers for people with schizophrenia. This is the top line result. We looked at self-reported cannabis use on the left and urinary carboxy THC on the right. What you can see there is in the active group, which is the open circles, on average about a 60% reduction in cannabis use that is on self-report, with a nice, medium to large effect size, little smaller effect with the carboxy THC. This was not creatinine corrected, so that is one of the limitations of this. But promising data in a very preliminary sample, just published in NPJ Schizophrenia, which is the journal of the Schizophrenia International Research Society. This is what we showed in terms of cannabis craving, particularly on appetitive craving. So what you can see there in the active group, in the open circles, is about a 30 to 40% reduction in craving over the course of the trial. And in that sham group, really no change. And the statistics were on the right side. We're certainly trending towards an increase. All right, so let me move on here. We also showed a nice effect on the positive symptoms of schizophrenia. Not on the negative symptoms. We showed about a 20% reduction in positive symptoms in the active versus the sham group. And I think, so here's cognition, a little bit complicated, but we showed in the active stimulation group an improvement in sustained attention on the CBT, and also some minor improvements on processing speed with the Trails A, and also some trends in some other areas like delay discount. That's a measure of behavioral economic measure of impulsivity. Finally, this was the tobacco findings. So of course, all these people were tobacco smokers, mostly cigarette, some vapors. But what we're able to show here is that with cannabis abstinence, or sorry, cannabis reduction, that the sham group actually showed this compensatory increase in tobacco use that we'd shown previously in Rachel's study. But in the active RTMS group, that compensatory compensatory tobacco increase was blunted. So again, I think trying to parse the interrelationships now between cannabis and tobacco using neuromodulation. So just in summary, in the last few minutes, in this preliminary study, we were able to show that reductions in cannabis were shown with high frequency RTMS. There was also a reduction in urine toxicology for THC, in cannabis craving, in the positive symptoms of psychosis, so delusions, thought disorder, hallucinations, a reduction in compensatory tobacco use increases, and an increase in some aspects of cognition, like attention. I won't go through all of this, but there were a number of strengths and weaknesses with this. We thought there was a pretty rigorous design, high retention rate, multiple modes of measuring cannabis use. Fairly rigorous design, but clearly statistically underpowered, and obviously the feasibility of doing RTMS, knowing that it's about $150,000 setup, it's probably not going to be in every clinic that we have, but we're bravely moving forward. This is the team we put together for the next series of studies. Larissa and Ziva, Andy Luchter, Steve Martyr are part of what we call Can Stimulate 2. We ideally wanted to do a big clinical trial across 4 sites, Harvard with Das Unger, Yale with Godfrey Perlson, and this is a picture of Larissa and I when she came to Toronto a few years ago. NIDA doesn't want us to do a big clinical trial. Can you guys hear me?
Yep, we can hear you.
Okay, good. Okay, I have one pod that's still working. That's good. But we're probably going to, move it into some mechanistic studies to get at, working mechanisms in terms of neuroimaging and neurophysiology that might explain RTMS effects that we saw. So stay tuned. Hopefully, we're going to have further discussions, and we're going to plan our next set of studies to follow up on this work. So I'm just going to skip that, and just tell you about one last interesting piece of data. This is my foray into survey and epidemiological research. This is Dave Hammond up in the top from the University of Waterloo, and Miriam, of course, is involved in everything. And the guy at the bottom is Wayne Hall, one of the leading drug policy experts from Australia. And this is from our International Cannabis Policy Study, which is a prospective evaluation of what are the health outcomes and other outcomes pre and post cannabis legalization in Canada. And if you look at people with different mental illness groups and daily cannabis use over the past 30 days, the only group that's changed in the first 2 years are people with schizophrenia or psychosis. Now, this is self-reported, self-defined, but it just shows that this population is actually very sensitive to policy changes. So all of Canada is now legalized. These were, I can't remember how many people, but maybe a couple of thousand people across Canada. And already we're showing changes in one very vulnerable group of cannabis users. So let me stop there. Whole bunch of collaborators that we're lucky to collaborate with in Toronto and all over the world. Here's a picture of everybody in the Center for Complex Interventions team. I wanted to thank David Castle, our director, who supported all this incredible group of young people. And thank you for your attention.
Great. Thank you so much, Dr. George. And with that, I'd like to open things up for questions. Let's see. I see Dr. Evans has his hand up, I think.
I did.
Yes.
Okay.
Hi, Chris.
Hi. How's it going, Tony?
Good. Nice to see you.
Yeah, a couple of questions. I mean, I'm kind of interested in the genetics of cannabis and schizophrenia. I mean, those that veil versus veil met seems a pretty pretty dramatic increase in the I mean, it's like a tenfold or 7 to 10, right? And then there's another one, that AKT gene seems to be very important as well. So you've got two of those. So my question, I guess, is, can you explain the heavy use just entirely on those two genes, the increase, or is it different?
Yeah. Well, you know better than me. I don't think, this is replicated so nicely when different people have looked at it. But no, I think there are multiple factors. I think cannabis use disorder, whether it's in schizophrenia, major depression, or not, is multifactorial. There are biopsychosocial determinants of that. There is, potency, there, availability. I think it's extremely complicated. I think, the early studies were very impressive. We want to look at this, in our samples. We have Jim Kennedy here, many other geneticists, but I don't know. We all thought maybe COMT, 23, we get it in 23andMe, and we have a
that's
adolescent psychosis prevention program. Hasn't quite worked out that way, has it?
Well, I don't know. I can't find my 23andMe veil. I would like to know if it's a veil met or a veil.
Absolutely.
I think it would be good to know, but you can't get that kind of granularity with 23andMe.
It's multi, it's polygenetic. Maybe Emma Johnson and Aparna Agrawal are looking at PRS scores. They're looking at pretty sophisticated GWAS. And they're using like a international data set to look at this. So I think there's a lot of really talented people that are looking at this comorbidity, and we just need to start genetics and everything else. We all need to start talking to one another, and because it's undoubtedly very complicated, the genetic associations.
Everything is.
Of course.
So you aren't doing genetics on that cohort with the TMS study is it? I guess, and I have one more question. Is there a difference?
We have DNA. I don't know what 19 people is going to make any difference, but
no, that's true. But is there any difference in the endophenotypes of the people who progress using high concentrations of cannabis to normal development of schizophrenia? No difference.
I don't think people have looked at it very well. It depends what kind of phenotype you're talking about. I think very relevant here would be cognitive endophenotypes, but I don't think we've got that kind of granular data. And so those are some of the big holes. That's why we were talking to Emma Johnson and Aparna Agrawal, because it may be our chance to apply complex genetic approaches to deep phenotyping data that we're obtaining. So if you have any thoughts on that, I'd love to talk to you more, Chris.
Well, I was really fascinated when you stopped the cannabis that the anhedonia became less and the depression became, you got rid of depression, but the depression got worse and the anhedonia got better, which is not real. It is sort of an opposite.
Well, no, the depression and the anhedonia got better in the same direction.
Okay, so
it's just it was more pronounced with the anhedonia. So there's a 50% reduction in depression scores and about an 80% reduction in anhedonia. So it's going in the same direction. So clearly, cannabis is not doing anything good for people with depression. In fact, 3 of those patients who quit were able to get off their antidepressants. So that's only 3 of 11, but clinically that was impressive. So
we do have one before we move on. I just want, with respect to the genetic, the genotype. So we do have one question from the audience that was, what's the prevalence of those COMT and AKT polymorphisms? So I want to make sure I get that question in before we move on too much.
So I believe it's what is the Val allele is 40% and the meta allele is 60%. So if you do the Hardy-Weinberg p squared 2p, you know, you can work it out. It's like, I can't remember, 16 and whatever, you know, it's 24 and 36 or whatever, you know, you can calculate the population prevalences if it's in Hardy. That's all I know about genetics. I got a B in genetics in undergrad, so that's why I try to deflect, you know.
And then going back to the depression and the anhedonia scores, it's really interesting how it did seem to improve over the 4 weeks. My question is, at baseline, were the participants abstinent, or, you know, were they like 12 hours abstinent, or had they just come in after they 12 hours?
The only thing we ensured is that they were not intoxicated, but they had pretty high levels of THC in their urine. But to do the baseline cognitive testing, all we said is we don't want them intoxicated. We didn't want them stoned, essentially. So we wanted them to be able to do the battery. And then we just let them be abstinent if they could the rest of the 28 days and proved it biochemically or not.
So is it possible, you know, I know we think about most withdrawal symptoms associated with cannabis use disorder, of course, take 1 day to 3 days to appear. But I'm wondering if you took this population pool and you looked at their depression and anhedonia 2 hours after they use cannabis or 4 hours after they use cannabis and looked at those scores, you know, would you see an immediate improvement of their depression and anhedonia right after they use cannabis, which might, you know, perpetuate this cycle of use?
So we didn't look at that at the really short timelines. I think we had a 4-hour one, just because it took 4 hours to do all the baseline workup and stuff, the cognitive testing, the clinical testing, and there was really no change because the withdrawal symptoms and craving were actually quite low, even if there were like 12 hours abstinent from cannabis. I don't know what it was done in a laboratory setting, but it wasn't rigorous. Probably it wasn't as rigorous as you would do, Ziva. You know, we had a little bit of flexibility there. Did we send them out for? No, we did not send them out for cannabis smoking breaks, even though we could have, but we didn't. So that's a good question. It actually would be really nice to do like a really nice, you know, hour by hour abstinence study. And in fact, Isabel Ballou is doing like overnight abstinence. So, and I have to ask her if it's in controls, but that would be a neat study to do, Ziva, to look at sort of, you know, these effects in clinical populations in that like a 12-hour window, like hourly or something like that.
Yeah. And actually we have a follow-up question from Dr. Chris Verico, who I'm sure you know. And he says, how did you ensure that no one was intoxicated in those studies at the baseline time point?
Sorry, how did you
ensure that no one was intoxicated at that baseline time point?
Clinical testing, finger, nose, heel, toe, good mental status exam. That was about it. I mean, we didn't have the fancy stuff that Eden Evans has with the you know, brain scans. But we had to determine it clinically. We actually got some training by one of those drug recognition officers and how to do that kind of stuff before we started these. So, but Chris, great question. Chris actually helped me with my very first experiments when I was a fellow back in the mid-90s at Yale. So it's good to hear from you, Chris.
Good to see you, Chris. And then another question, I know we have to wrap up soon, is, you know, it's interesting that in your recent study, you saw that cannabis craving went down in the active Rtms group, but it took a while.
It
wasn't until the 4th week until you actually saw the decrease in cannabis craving, yet participants self-reported that they were using less cannabis as soon as the first week. And I noticed that, you know, for tobacco, you saw decreases in tobacco use in the active group, but did you look at craving as well? I forget if you looked at tobacco craving to see if you saw a similar trend where where you would see decreases in use that preceded self-reported craving of that substance?
Ziva, that is a wonderful question. We have that data. You should look at it. That's actually a really good question. No, I'm pretty
Because
sure.
it raises the point with respect to how important is craving to the behavior.
Yeah, and certainly if we do this in a bigger study, whether it be a mechanistic study or a clinical trial, we need to do that right. We didn't have objective measures of tobacco like cotinine or anabasine. I'm pretty sure we have tobacco craving in that study, because so that's a good question. We'll go back and look at that, because all we're doing is we're reporting self-report. That's it. But I think those are good studies that will further parse what's going on if you look at that. So thank you.
Yeah, sure. Any last thoughts, Dr. George?
No, except stop calling me Dr. George. Tony, geez.
Well, it's wonderful to have you here. Thank you so much for
Thank
coming.
you.
And hopefully one day we'll be able to see you here in person.
Absolutely. Take care, everybody.
Thank you, everybody.